Hi.
Hi, Jennifer. How are you?
Good. Great to meet you
Yeah,
officially.
same. Um, do you go by Jennifer or Jennifer Lee or
Jennifer is fine. Yeah.
okay. All right.
Thank you.
Yeah, so you're a postdoc at Baylor, is that right?
Yes, I started in 2018, so it's been about 2 years.
Okay. Things
And
going
yeah,
well?
yes, things are going well. Applying for a lot of grants and trying to get research done. Like I said, Alzheimer's disease is a really small group in our lab. There's only 3 of us working on it, but it's fun. I enjoy it.
Good, good. Rick should hop on here any minute. I'm gonna rearrange here. I'll be right back, sorry.
Sure.
It's too bright outside. Sorry. Do you have a preference on days as far as, or do you think Friday will work for this?
Yeah, I'm open to any day of the week actually. Right now, my schedule is quite open and free. But Fridays at 3 work great for me as well.
Okay.
Hi, Rick.
Hey.
Hey man, how are you?
Good.
Good.
How are you guys?
Nice to meet you.
Same. Are you seeing me okay? Because I have to feel my internet is quite spotty.
No, you're
We
you're
can.
good.
Mm-hmm.
All right. So you guys already started discussing a bit about the dates. Did I overhear that?
We had just started talking about it.
Yeah. I'm open to any option really because my schedule is free. So.
I'm gonna try to log off and log back in.
Okay.
Okay. Since he's up, that wouldn't be good if his internet's not wanting to cooperate today.
I know. Some days I have really great Zoom meetings. Other days the connection is difficult and sketchy, but
yeah,
I guess that's what happens when everyone has to work from home and everyone's using Zoom to communicate.
Yeah.
Yeah, I think this is a great seminar series. I've advertised it to my lab and on Twitter, and I think it's great. I'm by myself here in Houston and I live by myself, so any opportunity to learn new things in the field or connect with other people in the field is great.
Yeah. I mean, I've been really pleased with how fast it took off and how willing everyone was to participate. So hope it keeps up.
Me too.
So assuming that Rick is also okay with Fridays at 3, if we go with that, is there anything holding us back from saying something today about it?
No, I think we should. We should definitely introduce it and tell them that we'll have like a Google sign-up sheet similar to what you did with the PIs, available on Twitter.
Yeah. Although I will say, I think we've got a couple postdocs who are in that. I didn't think about that till now. A few of those individuals who signed up are postdocs.
Or postdocs, I see.
Yeah. Hey, welcome back.
Yeah, I think this is better.
This is good.
So I was just saying, Rick, that I hadn't thought about this before, but a couple of the people who signed up in the regular sheet were postdocs.
Yeah. Fine, right?
I think that's okay.
Yeah.
I think we can leave it.
I think it's just if you're like a junior postdoc or like a senior postdoc, it makes a big difference also. I think some postdocs might not be comfortable with a 45-minute talk, but would be with a shorter talk, I think.
Yeah. Okay.
Yeah.
We'll just kind of frame it like that. Jennifer, do you want us to give the pitch, or would you rather do it yourself?
Maybe you can start and then introduce me, and then I can talk a little bit, introduce myself, and or talk a little bit about my background, maybe, and
Sure. Look,
along those lines.
yeah. Maybe a quick little thing about. So, like you said, there's a tab now on the on that same Google sheet. So the same link will work. We can use this same Zoom link for the webinar. Provided you good with that, Rick? You see what I saw in my email? Like we can either take turns, or I can just set it up, and then Jennifer can drive as a panel.
Yeah, that's fine. I think that's good.
Okay. So then sounds like we settled on Fridays at 3.
Yeah.
That works for me.
Okay.
I might not make all of them, but I think that's fine also.
Okay. Yeah. So we'll do that then. I can just quick welcome, and then throw it over to you, Jennifer, to pitch, for trainees, graduate students, junior postdocs, to please sign up. Same link. There's a separate tab on that Google Doc. You guys think we'll shoot for 2 15, 20-minute talks to start with?
Yeah, I think so.
Yeah.
Okay. All right. Well, I'll plan on doing that. I'll shoot it over to you, and you can give the sales pitch.
Sure.
Yeah, I don't think we have to sell it. It will sell itself, I think.
What's that, Rick?
I hope.
It will sell itself, I'm sure.
I think so.
Yeah.
I think it's a good opportunity.
Definitely.
Are you looking for like a co-organizer still, Jennifer? That would be nice, I think, also to have 2 people.
Yeah, I have somebody else in my lab that would be interested in co-hosting with me. So that'll make things a little bit easier to coordinate between the 2 of us, since we're in the same lab.
Yeah.
So yeah, I'm not sure if she'll be on today, but I wanted to talk to my PI today to discuss. I really would love to present my own work. I just want to make sure it's okay with her, and get her thoughts and opinions on it.
Yeah,
Sounds good.
And Lance, then go back to you, or should I then introduce the format again? I think so, right? I just like the format for our seminar tonight.
Yeah, that works. And then I'll introduce you.
Yeah, sounds good.
Okay.
Do you have a slide with the program? Are you going to rotate those same slides?
Okay, actually here, let me see if it'll let me share my screen with you guys.
Yeah.
I've got this. I've got this. I've got your beautiful mugshot. And then I did this. Do you guys want me to throw this up too?
Yeah.
Yeah, that works.
You should be a designer, man, not a scientist.
Anything you want me to add here or change here? Because I can do it. We got time.
Yeah, maybe I'll send you a better picture.
If you want me to change the picture or take it off, I can.
I'll send you a better one, but you can leave it for now.
Okay, I think that's a nice picture. I don't know what you're talking about.
Thank you.
All right, cool. So I'll let those run. I think I've got this set up to go automatically. Make sure.
And I think we forgot last time also to turn our pointer into the laser pointer. And I'm going to forget again. I already know if I do this, I'm going to forget it again. So if I don't switch it to the laser pointers, can you guys remind me of?
Okay.
Sure.
And then you're going to keep some time for me, Lance, also, right?
I will. I'll give you a warning. When do you want a warning? 35 minutes or so? 30 minutes? 40 minutes?
Yeah, 35 is good. I don't think I'll go over time, actually.
Okay. All right. What am I doing here? Slideshow. Transitions. That's what it is. Yep. Okay.
So where are you at, Jennifer?
I am at Baylor College of Medicine.
Oh, yeah. In
Houston, Texas. Yeah. Mm-hmm.
Yeah. Are you from Texas?
I am. I'm originally from El Paso, Texas. Okay. And my family still lives there. And so I moved to Houston for my PhD. So I did my PhD at Baylor in the lab of Mauro Costamarioli, looking at translational regulation of memory. And then I decided to switch to Alzheimer's disease. So I stayed in Houston and I joined the lab of Huda Zogby, where
Okay.
she studies a bunch of different things, including Rett syndrome, spinocerebellar ataxia, and now a small component of her lab also studies Alzheimer's disease and Parkinson's disease.
And when did you join the lab?
In 2018.
Okay, so
I've been there about 2 years. Mm-hmm.
Cool. Yeah, I haven't never been to Houston actually. I think it's like I've never been to Texas in general. I think I missed out on that, the South a bit when I was in the US.
Yeah, we'll have to send you an invite for a seminar.
There you go.
Yeah, that would be great. Yeah.
Yeah.
An online seminar.
An in-person seminar. An in-person seminar.
Yeah, I do collaborate a bit with Hugo Bellen. I think he's
Okay.
also over
Yeah.
there. I don't know if you guys are in the same department or.
Yes, we are. Yeah, and my boss Huda's very close to Hugo.
Yeah.
He's only a couple floors down from our lab, so.
Okay.
Yeah.
Yeah, he does cool stuff. Also, I'm gonna reference his work a little bit also in my talk.
Oh, great.
I'm gonna be right back, guys, and then I'll open this up in just a few minutes.
Sure.
Yep.
So should I also introduce myself a little bit, or should I just stick to the mentioning the happy hour for grad students and postdocs?
Why don't you let me introduce you real quick? I'll just say this is Jennifer Johnson. She's a postdoctoral fellow in the lab of Huda Zogby at Baylor, and she took the initiative to start us up here, and then I'll kick it over to you to describe how it will work and where people can sign up and all that.
Okay.
Yeah, Lance is a natural. Like he's like hosting this. Like he's been on TV for like many years.
Yeah,
People,
I agree.
Making transitions.
I don't know about that.
Did you get good feedback from your talk, Lance? Did you like speak to people afterwards, or?
I did. I got several emails afterwards. Actually, Jennifer, do you know Virginie? What's her last name? Brugia Breveau. I think she works with you guys. Maybe not your lab, but with Hugo's lab and some others at your institution. So yeah, I had a few people reach out, ask questions, wanted to chat about stuff. So it's been great.
Yeah. Cool. Don't forget to close the chat, by the way, before
So I was just checking on that. It says right now that no one can chat. So.
Yeah, the Zoom bombing is a thing now, right?
Yes.
Were you there for the first seminar, Jennifer?
When yeah,
Jacob,
I took a picture.
Oh yeah.
I took a picture of it. I'd never seen that before. And then I said, who still uses T-Mobile Sidekicks?
Yeah.
They make those anymore?
No, I don't know. But like that was the second talk. The first talk we actually had, we still had to chat. Like we gave someone the microphone and he started like moaning and making
oh my God.
Yeah, I should have known better.
Yeah, we should have known better. He was like already asking to get a question like from the start, and
yeah.
Wow.
Yeah, it says chat's disabled right now. So
yeah, it's a shame, but I don't think it's that's going to be some inappropriate stuff. The only thing that I'm worried about that people still might have the old link.
I saw Shane tweeted out the old one, which was a problem.
Again, or just like the last time you did?
Last time. So you know what I didn't do was I should have had it open on my phone with the new link. Don't know if I can be able to do that quick enough. Oh well, I don't know if I can figure that out. Next time I'll do that.
Yeah.
All right, I'm gonna go ahead and open this up, guys.
Okay.
All right, that screen share is working.
It is, yes.
Yeah.
Yeah, I heard from everybody that their PhD defense got Zoom bombed as well, but people just get in and
really,
and yeah, I think we're lucky because people can't they're muted when they come in. But like for a regular Zoom meeting for a PhD defense, everybody can just talk, and it's been horrendous. I heard from people that they got like trolls in there.
All right, welcome everybody joining us. We're going to wait a few minutes and let people trickle in before we get started.
It's a good picture of me, I have to say. They photoshopped that, I think.
I thought it was a nice one too.
Yeah, I think it was like a fundraiser that they made it for. Yeah, it got me. I didn't get the funds. I got the picture though, so it was a very good investment also.
It's pretty good.
Yeah. Have you guys been following ADPD?
I was on there a little bit today. Yeah, some good stuff.
Yeah, I saw that they presented Diane at TU results, right?
So yeah, I haven't. That's on my list to watch. I haven't done that yet.
So you're registered for the meeting?
Yeah.
Oh, that's good.
I was a little shocked at how expensive registration is for a virtual meeting, but
yeah, I think they had everything organized and then they had to switch it to a virtual conference.
Right.
But it seems like a very good program. They also had like a whole APOE section in there.
All right, guys, for those of you just joining us, we're going to give this about one more minute and then we'll get started. Rick, we didn't try your slides yet, did we? But I'm assuming it'll be okay.
Yeah, I have the dual screen setup over here.
Okay.
I feel very prepared for screen sharing.
Okay.
Don't forget to tell me to switch my pointer to the laser pointer.
Okay.
I am gonna forget, I'm sure.
All right. How about we get going?
sure.
Okay. Welcome back, everybody. A few things to start off with. I'd like to thank everyone on Twitter and otherwise who has helped spread the word for this seminar series. It's really taken off, and I've been really excited Rick and I have been really excited to see how far this has gone. Thank you to Elizabeth Wu at Alzforum for helping spread the word, as well as Heather Snyder at the Alzheimer's Association. Before I introduce today's speaker, Rick Vanderkant, I'm going to actually kick this over to Jennifer Johnson, no relation as far as I know. So Jennifer has agreed to organize a new happy hour trainee talk session. So Jennifer is a postdoc in the lab of Huda Zogby at Baylor. And as I said, she's going to take over the organization for this new trainee specific seminar series. So Jennifer, go ahead.
Great. Thank you, Lance, for the introduction. Yeah, I'm really excited to host this happy hour seminar talk. And for those grad students and junior postdocs that may be interested in signing up, there's going to be a separate tab in the original Google sign-up sheet where you can sign up with dates that are good for you. It's going to be on Fridays at 3 p.m. And the Zoom link will be the same as the Zoom link for Tuesdays and Thursday seminars. You can also contact me with any questions that you may have. And my email, I believe, is on, it shows up on one of these slides. It's jljohnso@bcm.edu.
There you go. Okay, great. Thanks, Jennifer.
Thank you.
Okay, so it's my pleasure to introduce today's speaker, Rick Vanderkant, my co-host and new electronic friend. So Rick got his PhD at the Netherlands Cancer Institute in Amsterdam. There he was studying the interaction between cholesterol metabolism and endosomal transport. Then in 2013, he took off for San Diego to do a postdoc in the lab of Larry Goldstein at UCSD. And so, of course, Larry's lab was really one of the first to begin to apply iPSC technology to AD, and Rick was a big part of that. And then as part of his European Research Council fellowship, Rick returned to the Netherlands in 2017 as an assistant professor at the VU University in Amsterdam and the VU Medical Center in Amsterdam. And so his lab is really interested in understanding the cell biology, the cell biological processes underlying AD, and developing new drugs that can target some of these early pathological pathways in the disease. And so, Rick, I'm excited to hear your talk, and the floor is yours.
Thank you very much. And I'm very excited to give this talk. Feels a bit like the commentator has to go into the pitch and play his game now, so I'll try to live up to that. Let me try to share my screen. Yes. Do you guys see my screen now? Is it in the...
I think you're good to go.
Laser pointer.
Exactly, laser color, red.
Thank you.
There we go. Awesome. All right. So today I'll talk about how cholesterol metabolism is actually a dual driver of neuronal Alzheimer's disease pathology. And I know that a lot of people in this seminar series are actually interested in astrocytes and microglia and ApoE. And of course, I will not keep those out of this talk either. So I'll focus firstly on neuronal cholesterol metabolism. That's the work I've been doing so far. But then later I'll kick in some astrocytes and microglia and talk about the things that we're doing now in my lab. So as Lance already introduced me, my lab is interested in understanding how cell biological pathways downstream of genetic risk factors drive sporadic Alzheimer's disease. We study these pathways by combining induced pluripotent stem cell models, and we also combine that with gene editing to study risk variants in human neurons, astrocytes, and microglia. And our goal is to develop novel targeted therapies that can halt or cure Alzheimer's disease or related dementias. So I'm assuming that induced pluripotent stem cell technology does not need a lot of introduction. So just a short overview of that. So this is a technique that was awarded a Nobel Prize a couple of years ago, by which you can take any cell of the human body and turn that cell into a stem cell. And this is called an induced pluripotent stem cell. The nice thing about these stem cells is that they can make almost any cell type out of the body with the differentiation protocols. And for our interest, we can make neurons, we can make astrocytes, and we can make microglia of these iPSCs. So mostly focus my talk on the neurons today, as I said, but I'll also talk about co-culture models. It's a bit more of an advanced model for using iPSCs to study Alzheimer's disease. So something that's traditionally often done is taking iPSC cells from a control or from an AD patient and comparing these two patients for phenotypes. Something that we, or most people start doing more recently, is actually using these iPSCs to do gene editing in and introduce, for example, risk variants for Alzheimer's disease. And this is called an isogenic background. All the genes are the same, all the variants are the same, except for the one position where you're interested in. So as Lance mentioned, I did my postdoc in the lab of Larry Goldstein, and he was one of the first people to actually apply this iPSC technology to Alzheimer's disease. And this was a paper published before I actually joined the lab in 2012. So in this paper, it was published in Nature. They took iPSCs from 2 non-demented controls, 2 sporadic AD patients, and 2 familial AD patients. And these patients have an extra copy of the APP gene. So they develop very early onset Alzheimer's disease because they overproduce A beta. When we derive iPSCs from these patients and we derive iPSC-derived neurons, we can measure AD pathologies in a dish. And that's depicted over here. So we measured, or they measured, A beta secretion from these cells. And you can see that A beta secretion is significantly higher from these patients with this extra copy of APP, as you might also expect, of course. In addition, one of the sporadic AD patients also showed higher A beta production, whereas the other one did not. That's of course interesting, but maybe not that exciting, because we know actually already that these have an extra copy of APP. What I think is really exciting though is that they also have increased levels of phospho-tau. similar as the patients have in vivo, there's an increased phosphorylation of tau species. And again, that's seen in both of the APP duplication patients and one of the sporadic AD patients, but not the other one. So when I joined the lab in, I think it was 2013, a lot of the amyloid targeting therapies were already hitting the clinical trials. This space was very well covered, so instead of focusing on A-beta, what we actually decided to do is actually focus on tau phosphorylation. And what we decided to do is do a drug screen to identify drugs that reduce tau hyperphosphorylation in this APP duplication iPSC-derived neurons. So we took this one patient that had the highest level of phosphorylated tau, and we did a phenotypic drug screen in this iPSC-derived neurons with a library of over 1,600 compounds that were all FDA or EMEA approved and preclinical drugs. So drugs that are actually ready to go into the clinic. So this is the protocol that we use for this drug screen. We derive neuroprogenitor cells from these iPSCs, and then we differentiate them to neurons, and we call them differentiated neuroprogenitor cells because there are still some non-neuronal cells in there as well. So 90% of these cells in these cultures are neurons. A small fraction is astrocytes, neuroprogenitor cells, and some other cell types that we cannot really identify. So after 3 weeks of differentiations, these look like functional neurons, and then we can replate them into screening well formats. So a 384 well plate, for example. And what we did a drug screening to find compounds that actually reduce the tau hyperphosphorylation in these iPSC-derived neurons. And we did that as a measure by ELISA, and we also did a measure of cell viability. And I have to say, all of this screening work was actually done in collaboration with Anne Bang at the Sanford Burnham Prebys Institute, also in San Diego. So we screened over 1,600 compounds, and I say we, but it's really Anne and Cheryl Herrera, technician in the lab. And this is what this screen looked like. This was months of works just shown on this one figure. So here are all the DMSO controls. On this axis is the effect of tau phosphorylation. And in pink are all the compounds that we screened in duplicate. And what we're looking for is compounds that reduce tau phosphorylation. So compounds that show up on this lower part of the graph over here. So of the over 1,600 compounds that we actually screened, we could identify almost 160 compounds that gave a hit in our screen. So they showed up as reducing tau hyperphosphorylation. And we rescreened all of these compounds. And then we ended up with a compound library of 42 compounds that did reduce tau hyperphosphorylation in 2 duplicate repeats and 2 duplicate assays. And we confirmed those as non-toxic hits that reduce tau hyperphosphorylation. So what are these hits? And this leads me to the title of the talk, also cholesterol metabolism. As you can see over here, we had 42 compounds. This is the effect on tau here. So the bluer these are, the more the effect they have on tau hyperphosphorylation. And on top here is the viability of the cells. And what you can notice is that there's different groups of compounds. There's compounds that interact with microtubules, which is nice because tau is a microtubule binding protein. So this is something that you might actually expect for these drugs to affect tau hyperphosphorylation. There's a group of cholesterol targeting drugs, and I'll come back to those drugs extensively. There's a group of drugs that regulate neurotransmission. There's a group of drugs that interact with DNA, estrogen receptor signaling. And there's a group of drugs that we don't really know what they do. So we place them in this miscellaneous group. So this work has been published in 2019. So if you're interested in any of these drugs, please feel free to go back to that paper and let me all know also if you're interested in following up in one of these drugs. So what we decided to focus on is this group of drugs. So these are the statins, and many people know statins. Statins are the most widely used drugs in the world, and they inhibit cholesterol synthesis. And what we noticed that all statins in our drug screen actually popped up as hits that reduced the tau hyperphosphorylation. And that's something that we were very interested in. As I also mentioned, I did my PhD working on cholesterol metabolism. So this is something that immediately drew me to these hits. And also the fact that statins have been shown actually to reduce the incidence of Alzheimer's disease in large studies as well. So we wanted to go into this mechanism a little bit more of statins. So what we did first is we ordered all the statins that we could order, and we did the dose response experiment, where this is the tau phosphorylation over here, and these are increasing doses of statins. And what you can clearly see is that with increasing doses of statins, tau hyperphosphorylation is reduced. And this has no effect on cell viability of these iPSC-derived neurons. As depicted over here in black is the effect on tau phosphorylation. It goes down, but the neurons in red, the viability is not affected at all. So these neurons really tolerate these statins well, and they reduce tau phosphorylation in a dose-dependent manner. So this is not only true for just one tau phosphorylation epitope. So what we did our ELISA screening for was this tau phosphorylation epitope at threonine 231. We also confirmed that this effect was also true for other tau epitopes, such as this 396404 phosphorylation epitope, which is here in the back of tau, but also for the 202 and the 205 epitope in the middle of tau protein. So that's shown over here. Demeso-treated cells have this hyperphosphorylated tau species. Atorvastatin-treated cells, it's almost gone from these cells. And over here you can see that these cells are still very, very viable. They look happy. They have nice axons and nice extensions. And this is where we're gonna go a bit deeper into the cholesterol metabolism arm as promised. So people that are familiar with statins, they actually might know that these drugs inhibit a very upstream step in cholesterol synthesis, which is this enzyme called AMG-CoA reductase. And AMG-CoA reductase is the first enzyme that converts AMG-CoA to mevalonate. And mevalonate is a precursor to cholesterol, but it's also a precursor to other important cellular metabolites such as geranyl and proteonyl farnesylation. So to really find out which of this pathway actually mediated the effect of these statins on tau hyperphosphorylation, we really dove into this pathway, and we used different inhibitors that inhibit different parts of this pathway. So what you can see over here is that we measure tau phosphorylation again by ELISA. This is the DMSO control. This is the atorvastatin, a strong decrease. If we now use this inhibitor, which inhibits protein farnesylation, we do not see any effect on tau phosphorylation. If we use a geranyl transferase inhibitor, also no effect on tau phosphorylation. So indicating that these two branching pathways do not really affect tau hyperphosphorylation in these cells. If we go down a bit more into this pathway, looking at the cholesterol-specific arm of this pathway, we use an inhibitor of the squalene synthase. We do again see a significant reduction in tau hyperphosphorylation. And also with a more distal inhibitor for DHCR7, which inhibits the almost last step to cholesterol conversion, you also see again a significant reduction in this tau hyperphosphorylation. So this really indicates that it's cholesterol that's mediating the effect of statins on tau phosphorylation. Well, as you know, you can reduce cholesterol metabolism in a number of ways by inhibiting its synthesis, but also inducing its export. And this is a process in which ApoE is actually also involved. Cholesterol is regulated by this liver X receptor. If you activate this liver X receptor, you induce the export of cholesterol out of the cell in an ApoE-dependent manner. So when we activate this liver X receptor with an agonist, we again see a very strong reduction of tau hyperphosphorylation in these iPSC-derived neurons. Another way to induce cholesterol export is by activating this enzyme, CYP64A1. And I'll come to talk about this enzyme a bit more later on in my talk. It's a neuron-specific enzyme which converts cholesterol to 24-hydroxycholesterol. And this 24-hydroxycholesterol is soluble and it's actually released from the cells. So again, if we give this effervescence to our iPSC-derived neurons, we see a very strong reduction in this tau hyperphosphorylation. Again, indicating it's really cholesterol that's driving this phenotype, as summarized over here. So what we did as good scientists, of course, we wanted to validate that indeed that the compounds that we tested in our assays actually affected cholesterol levels in these cells. So this is the summary of a couple of compounds, their effect on p-tau, and we wanted to see how well that correlated with the effect of these compounds on cholesterol levels in the cells. And this is what we saw when they actually measured cholesterol levels. Almost no change in cholesterol levels with any of these compounds. And the only change that we actually did observe was this compound, which did reduce cholesterol levels, but it almost did not affect tau phosphorylation. So it was one of the weaker hits that we got from our drug screening assay. So it was very puzzling to us. So we have all these inhibitors that's supposed to be reducing cholesterol levels, but they don't show any effect on cholesterol in our cells. And that's when we decided to dive into the lipids a bit better. This is a mass spec of cholesterol and related compounds. And what we were aiming to see is if look at these precursor molecules, for example, that's mosterol, 7-DHC, to really check if these compounds were really doing what they were supposed to be doing, which is inhibiting the synthesis of cholesterol. And they did. We actually showed that all of the compounds were behaving as we expected them to behave. But again, this puzzling result that they did not produce cholesterol levels. And then we noticed this small peak over here. So we even decreased the cholesterol peak over here. It's really a high peak. This is really a small peak over here. But we had the idea that we saw some differences in that region of the lipid mass spec results. And these lipids over here are actually not cholesterol, but they're cholesterol esters. And I'll come to speak about those quite a bit more in my talk. So we found a collaborator that was capable of measuring these cholesterol esters. And what we then observed is very striking, that these cholesterol esters were actually decreased in a fashion that's very similar to the effect of these compounds on tau hyperphosphorylation. So this is really indicating that it's not cholesterol that's being changed by these molecules, but it's the cholesterol esters, and that these cholesterol esters then have a direct effect on tau phosphorylation. So we also tested that by using ACAT inhibitors. So ACAT is the enzyme that actually converts cholesterol to these cholesterol esters. And we also showed that giving cells these ACAT inhibitors to prevent cholesterol esterification, we also again saw a very strong reduction in tau hyperphosphorylation. Again, indicating that it's the cholesterol esters that have an effect on tau phosphorylation, as depicted over here. So this was of course an interesting observation, and we were very interested in following up to discover how this pathway was regulated. And the first thing, of course, that we thought about was looking at amyloid as well, because we know that amyloid can be a regulator of tau pathology. So what we found is that these cholesterol esters directly regulate tau phosphorylation. What other people have previously shown, including a lot of work from Dora Kovac, is that these cholesterol esters also regulate the processing of APP to A beta. And that's also something that we observed in our iPSC-derived neurons. When we treat these neurons with atorvastatin, you see a very strong reduction in A beta production as well, also indicating that statins regulate A beta generation in our system. And when we correlated changes in A beta to changes in this phosphorylation of tau with different concentrations of the drugs, and with different times of treatments, we saw this very strong correlation between A beta and phospho-tau. So if A beta would go down by 50%, phospho-tau would also go down by 50%. So this indicates a correlation or maybe even a causal relationship between A beta and tau in this system. And there's, I think, two different ways that you can explain this data. One is by the amyloid cascade model, in which cholesterol esters have an effect on A beta production. And then this A beta has a direct effect on tau phosphorylation. There's an alternative way of thinking about this cascade, in which these cholesterol esters drive both A beta, they drive p-tau, but in an independent manner. So they affect both pathways, but not in a causal way. So how we decided to test this hypothesis, or to decide which of the models describe this pathway best, we use CRISPR-Cas9 gene editing. So as I mentioned, we have these APP duplication patients, iPSC-derived neurons, so they have an extra copy of APP. And what we decided to do is by CRISPR-Cas9 delete all 3 copies of APP in these cells. And this indicates that you can get a completely knockout cell line for APP, and the residual signal over here that you see is actually not APP, but it's cross-reactivity of APP antibody with this APLP2 protein. So APP is completely gone from these cells, and A beta is completely gone from these cells as well. So the question then is, what now happens to tau phosphorylation if we treat these cells with statins? And this is again showing you the plot for the tau phosphorylation in the control cell lines, so the APP duplication patient. With increasing doses of statins, we see dose responsive decrease in tau hyperphosphorylation in this APP duplication page. So now we tried the same in this APP knockout cell line. And of course, you would expect, if the left cascade is true, that there's no more effect of these statins on this tau hyperphosphorylation if APP is required to mediate this effect. However, this is what we discovered. These cells completely had the same response to statins on tau phosphorylation, indicating that the effect of statins or any cholesterol-lowering drugs on tau phosphorylation is not mediated by APP or by A-beta. So that's one way we showed that this is not a cascading model, but a dual upstream regulator model. This is the second way that we also went about it. This is a very elegant paper published in 2012 by Barrett et al. And what they actually showed is that APP has a cholesterol binding domain, and they showed that by crystal structure that the C99 fragment of APP can actually bind cholesterol. And they also identified some crucial amino acids in the APP molecule that actually bind to cholesterol. So what we decided to do, again, by using CRISPR, and this was in collaboration with a very talented PhD student, Vanessa Langnes, we decided to introduce point mutations in APP that would abrogate the binding of APP to cholesterol. So this APP can no longer bind cholesterol after we made these mutations. And this is then what we observed if we treated these cells with statins. So these are the wild type cells. If we treat these cells with statins, we see a reduction in secreted A-beta. In the now mutant cell lines, if we treat these mutant cell lines with statins, we no longer see that A-beta production is responsive to changes in cholesterol levels. So what happens to tau phosphorylation in these cells? So again, this is the control cell line. Giving these control cells statins reduces tau hyperphosphorylation, as we also previously saw, but also in this APP cholesterol mutant cell line, this reduction of cholesterol levels still reduce tau phosphorylation, again uncoupling the regulation of A-beta and p-tau by cholesterol in the system. And we think this really indicates that cholesterol esters are actually dual upstream regulators of A-beta and tau separately. So it's not a cascade. It's a dual upstream regulator model. All right, so then the next question is, if it's not A-beta, what is it then that regulates this effect of cholesterol esters on p-tau? And what we decided to do is look at multiple phosphorylation epitopes by Western blot. And we saw that not only were the different phosphorylation epitopes of tau reduced by Western blot, but these different cholesterol-lowering drugs, also the levels of total tau were actually being reduced by these treatments. And this is indicative of maybe a proteostatic event rather than an event regulated by phosphorylation or dephosphorylation. In the next experiment, we try to rescue the effect of reducing these cholesterol esters in these cells on tau phosphorylation. So again, this is tau phosphorylation on this axis here. If we give these cells statins, tau phosphorylation goes down. And we try to rescue this effect by either inhibiting autophagy, inhibiting phosphorylation, or inhibiting proteasomal degradation. And in this case, what you can observe that we could not rescue this reduction in tau by either inhibition of autophagy or phosphorylation, but we did inhibit the reduction in tau by blocking the proteasome. So really indicating that this effect of statins on the tau phosphorylation is dependent on the proteasome. At the same time, we had also done a proteomic study of the cells after treatment with statins. And there we also saw that there was an upregulation of certain proteasomal subunits. And that's also something that we could confirm by Western blot. It's quantified over here. There's a couple of proteasomal subunits that are actually increased after we do statin treatment. Again, indicating that somehow the proteasome is affected by cholesterol ester levels. So to further confirm them, we used this very elegant system that was developed by Hype Ova at the Leiden University, also here in the Netherlands, where they developed an activity proteasome binding probe. So these are chemical probes that are fluorescent, and they can enter the proteasome. And when the proteasome is active, these compounds covalently bind to the proteasome. And you can then quantify the amount of fluorescent proteasome either by Fox or by Western blot, as shown over here. So we also did that assay to see if we really had an activation of the proteasome by lowering cholesterol ester levels. And again, that's something that we observed in 2 different cell lines. So this is a control cell line. This is the APP duplication patient cell line. If we give these cells statins, or if we reduce cholesterol levels by efavirenz, we see an upregulation of proteasomal activity. Again, indicating that these cholesterol esters are somehow regulating the proteasome in the system. So that data is summarized over here. That's the conclusion we made for the cell stem cell paper that I published in 2019 also. So cholesterol is over here. Cholesterol can be stored as cholesterol esters. If you either inhibit the production of cholesterol or you induce the export of cholesterol, these cholesterol esters are converted back to cholesterol, thereby the levels of cholesterol esters go down in the cell. And through 2 independent pathways, one by affecting A beta and one pathway affecting p-tau, we can see a reduction in both p-tau and A beta if we reduce these cholesterol ester levels. All right, so next I'm going to talking about some movement to the clinic that we're actually making, and I showed some happy doctors over here because this is normally where the clinicians get excited. I'm afraid there's not too many happy clinicians at this point, but maybe this will make them a bit happier. So when we talk about cholesterol metabolism in Alzheimer's disease, we often get the comment, so why are you even probing cholesterol metabolism? Because we already know the statins don't work. And that's true. There has been some clinical trials with statins, and they actually did not show any effects on cognitive decline in patients. They could not rescue these cognitive decline in patients. However, most statins are actually quite poorly brain permeable. And even the brain permeable statins, such as simvastatin, they don't really reach high enough concentrations in the brain. In addition, clinical trials with statins have been performed in mild to moderate AD patients and often excluded subjects with high circulating cholesterol. And I think if we go back with the knowledge we have now about Alzheimer's disease, this would be a very late treatment stage for these patients. So those statins have not been really evaluated in the earlier AD cases. And last, statins are very pleiotropic drugs. They do not only inhibit cholesterol synthesis, they also inhibit these branching pathways over here. And that's potentially very toxic, possibly also for astrocytes, as I will show on my next slide. So we think that this efavirenz is a much better drug than these statins. Efavirenz is highly brain permeable. It's specific to cholesterol, so it really affects the later part of these pathways. And it targets a neuron-specific enzyme. So CYP64A1 is only expressed in the CNS. So it's a very clean way of modulating cholesterol metabolism in the brain. And in addition, this 24-hydroxycholesterol is actually secreted from the brain into the blood and can be used as a target engagement marker to see if the drugs are really getting to the place where they have to go and whether they're activating the target. So as I said, we really like this efavirenz. And one of the reasons is that we also showed in our paper is that this efavirenz is not toxic to astrocytes. So what we did in this case, we used iPSCs to make astrocytes. And then we again treated these cells with either atorvastatin, simvastatin, or efavirenz. And what you can see over here is that these neurons don't care about these statins. They're quite alive, they're happy. But astrocytes are really poorly tolerating these higher doses of statins. So at doses where actually these statins start producing tau hyperphosphorylation in neurons, these astrocytes start dying quite efficiently. So that's of course something that you do not want to have in your brain when you treat people. If you look at this graph over here, efavirenz did not have this effect. It's not acting on these astrocytes, and these astrocytes tolerated these drugs quite well. Tolerated efavirenz very well. So this has all been done in an iPSC model of Alzheimer's disease. And of course, before you go into the clinic, you would like to see something of this being repeated in an actual mouse, an in vivo mouse model. So that's what we did in collaboration with Robert Risman, also at UCSD. We did a chronic efavirenz treatment in the P301S tau transgenic mouse model. So we treated these mice for 3 months with efavirenz. And then we did a staining for PHF1, which stains phosphorylated tau. You can see the staining in different brain areas here on the left. These dark spots here are the phosphorylated tau that you can see by the staining. After efavirenz treatments for 3 months, we see a very strong reduction in this tau phosphorylation species in these mice. So indicating this is not only something that happens in iPSC systems, this is also something that we can recapitulate in vivo as well. And I think I cannot mention this work on effervescence without also mentioning the work by Irina Pukeleva. She was actually the first to show that this effervescence activates CYP64A1, and she has later also shown that effervescence reduces amyloid load in young APP mice and improves cognition also in late APP mice. So she has really worked on this amyloid part of the disease, and I think both our data are now indicating that this effervescence is really effective in reducing tau pathology, but also in reducing amyloid pathology. And this has led us to now think about clinical trials, and I'm very lucky to be at the VUMC Alzheimer Center, which is led by Philip Scheltens, one of the world leading clinical researchers in Alzheimer's disease. So we're going to work towards a clinical trial for effervescence. We're also very interested to test, of course, effervescence in other mouse models. So I'll be sure to actually add this compound to the compound submission portal that Garrett talked about yesterday in the AD model consortium. And also, unfortunately for the clinicians, I will go back to cell biology right now, and I hope you won't be too sad about that. So this is the mechanism that we cooked up in our paper. So cholesterol esters driving polysomal degradation of phospho-tau and A-beta. And I want to explain a bit more about these cholesterol esters. What are they and what are they doing in the cell? So cholesterol is normally stored in cells as free cholesterol. So it's not stored normally in cells. You have free cholesterol, which is all the cholesterol in membranes everywhere. If there's too much cholesterol in cells, this becomes very toxic to the cells. And they actually have to store it in these lipid droplets. And they do that by conjugating a fatty acid to cholesterol. And this fatty acid together with cholesterol is called a cholesterol ester. And these cholesterol esters are the main constituents of lipid droplet particles in the cells. And these lipid droplets do not only contain these cholesterol esters, they also contain triglycerols. So if we think that these cholesterol esters can actually drive Alzheimer's pathology, is there any indication to believe that these cholesterol esters or lipid droplets accumulate in the AD brain? And the answer to that, of course, is yes. Otherwise, I would not show you that slide. And I think this is something that Lance already showed also in his presentation. So this is a very early description of Alzheimer's disease by Dr. Alzheimer's himself. He describes the tau tangles, of course. He described the amyloid plaques. But he also describes that the glia have developed numerous fibers, and many glia cells show these adipose saccules. And these adipose saccules, I think, is something also that we now consider to be lipid droplets that might actually be full with these cholesterol esters. So this is a very old study, a relatively newer study in 2012, also showed by lipid mass spec that specifically cholesterol esters are increased in the brain of sporadic AD patients. And I also have to mention a lot of the familial AD mouse models, these cholesterol esters also accumulate. So indicating that this might be a broader pathway in Alzheimer's disease. So to answer that question, do cholesterol esters accumulate in Alzheimer's disease? The answer seems to be yes. The next question then is, actually what drives the accumulation of these cholesterol esters in the sporadic AD brain? And I'll talk a bit about the work that we're doing now to test if genetic risk variants in cholesterol regulating genes drive this accumulation of these cholesterol esters. And another question you could ask, which is really relevant for the treatment, is in which cell types do these cholesterol esters actually accumulate? And if you've been paying any attention to the recent literature, there were a couple of really exciting papers that actually addressed this question. So where do these lipid droplets and where do these cholesterol esters accumulate? So there's been 2 recent reports that show that these cholesterol esters can accumulate in microglia. One report by a group in Denali, and one report by the Tony Weiss-Correa group, I think they're in San Francisco. They show that under certain conditions, TREM2 and TREM2 dependent may these cholesterol esters accumulate actually in microglia. Also, Tony Weiss showed that these lipid droplets accumulate in microglia as well, although he did not show accumulation of cholesterol esters, but of other components of these lipid droplets. Do they also accumulate in astrocytes? I think that's also a question that Lance already addressed. So he shows that actually in ApoE4 mice, fatty acids and lipid droplets accumulate, which might be indicative also there that there's something wrong in the lipid metabolism in these astrocytes. And I think also a very cool paper that's on bioarchives is by Julia TCW. She will be presenting later in this series as well. And she also shows that cholesterol metabolism is deregulated in ApoE4 astrocytes. So what about neurons then? And the dogma is that neurons don't really produce many lipids, but there's a couple of papers recently showing that lipid synthesis increases under conditions of oxidative stress, mitochondrial dysfunction, and neuronal hyperactivity. However, neurons are not very efficient in making lipid droplets. So that could mean two different things. So maybe lipids do not accumulate in neurons. That's one way of thinking about this. Or another way that I like more is that these lipids actually accumulate in the neurons, but outside of lipid droplets. And that makes the accumulation of these lipids more harmful even for neurons than for other cell types. So we really want to address the question to whereas do these lipids accumulate in sporadic Alzheimer's disease? And one way about going about this is this, where we want to generate a sporadic Alzheimer's disease neurolipid atlas. So what we intend to do and what we're doing already is generate different sporadic AD risk polymorphisms and gene edit them by CRISPR-Cas9 in our iPSC system, creating an isogenic sets of cells, and differentiate these cells towards neurons, astrocytes, microglia, or co-culture systems. So now when we have these cells, we run them to an unbiased lipidomics assay by the lipidizer, and then we hope to get different lipid profiles in a cell-specific manner. So we can see how our lipids are changed in neurons, downstream of, for example, an ApoE4 risk genotype, in microglia, in astrocytes, and in co-cultures as well. And so we hope to get a sort of a map which will show a lipidomic cell type specific signature of sporadic Alzheimer's disease. And we think that can be very useful to target discovery. It can also be used as a drug testing platform. So we can evaluate, for example, drugs that target risk factors or lipid targeting drugs such as evavirans to see if they can really rescue these changes in the lipidome of these cells. And I think we think it can really help by identification of common or diverging pathways and phenotype between different sporadic Alzheimer's disease genotypes. So do they all converge on the same lipids? Do they affect different lipids, or do some of these risk genotypes do not affect lipids at all? And that will really aid in the cell biology of Alzheimer's disease risk variants. So I wish I could show you the data that we had, but we only did one repeat so far, and we really had the second repeats ready, but they are in the freezer now, and unfortunately, through this current situation with Corona, we don't have the data yet. But we started with the ApoE CRISPR, so we have ApoE3, 3, 4, 4, and 2, 2, iPSC-derived neurons and astrocytes, for which we have done the lipidomics. That's a PhD student in my lab that's working on that. We're also collaborating with Jessica Young, who's an assistant professor at the University of Washington in Seattle, and SORLA. So we have some very preliminary data on SORLA as well. And then we have a team of people working in my lab on TREM2, ABCA7, and also PLCG2, protective variant of BCL2 that protects from Alzheimer's disease. So we really hope to get a comprehensive map of the lipidome in Alzheimer's disease. And we're very much interested in also collaborating with other people that are interested in different genes or are studying the same genes but in a different system. And I have to make one major acknowledgement to my lipidomics collaborator, which is Martin Vieira, also at Leiden University in the Netherlands. And he's been really the lipid wizard behind this project. So what we also want to do is compare these lipid changes to that we see in our iPSC-derived models, compare them to human tissue and also mouse brain tissue. And so we have funding to do this for ApoE4 and ApoE3, but other mouse models will be of high interest to us. And for example, ABCA7 or TREM2 models will be very interesting for us to test. Also, their collaboration is very welcome. So in the last 5 minutes, I really want to go into this co-culture system there. Am I still good on time, Lance, or do I have to rush?
You're okay. I'll give you 5.
Excellent. So I mentioned that we're going to measure all of these lipids in the individual cell lines, but I also want to highlight why I think it's really important to look at co-culture systems as well. And the reason for that is that glial genes seem to regulate neural cholesterol metabolism and vice versa as well. So there's a bit of the dogma in the field. So we all think that astrocytes actually produce lipids, and they send these lipids by means of ApoE to the neuron. I think that's a couple of really cool papers that came out over the last years, 2 Cell papers, that actually showed that the opposite can also happen under certain conditions. So under conditions of reaction to an oxygen species, mitochondrial respiration inhibition, or hyperactivity of neurons, these neurons now also start producing lipids in quite high numbers. And what happens then is that these lipids are then transported to astrocytes where they're actually stored. So this is something that I'm very interested in. So now suddenly we have a complete opposite observation where neurons can also produce lipids and send them to the astrocyte. So we also showed that in our iPSC-derived neurons that the similar system is also working. So if we impair mitochondrial respiration by drugs in our system, we can see a very strong increase in, we see an increase in cholesterol esterification in these cells. So the orange and the gray over here are 2 different concentrations of the same drugs. And we see an increase in lipids that also are in these lipid droplets, the triglycerols. And those are both components of these lipid droplets that are increased if we inhibit mitochondrial respiration in these cells. So we can also show that these lipids that are generated in neurons can be transported to human astrocytes. So this is a system that Iris Rosmalen in my lab developed. When we co-culture these astrocytes alone, we don't see too many lipid droplets. However, if we now co-culture them with neurons, we can suddenly see that a lot of these lipid droplets seem to be occurring in these astrocytes. So really indicating that these neurons are sending lipids to the astrocytes. And so the same experiment is depicted over here. So these are astrocytes that are co-cultured with neurons. You can see an accumulation of lipids in these astrocytes. You don't see the lipids accumulating in monocultures of these astrocytes. And when we now induce mitochondrial respiration stress, we can see that in the neurons specifically, we see a very strong enhancement of this signal. So these neurons make more lipids, they send more lipids to these astrocytes as well. So another way of showing that is also something that we're doing in the lab is by loading neurons up with fluorescent cholesterol, for example, in this example. We load them only in the neurons, then we co-culture these neurons with the astrocytes, we separate them again, and then we can very clearly see that somehow this cholesterol that was only given to the neurons now ends up in the astrocytes as well, as you can see over here. Again, indicating that these neurons can send lipids to the astrocytes. And I think the very nice thing is that we can do this by microscopy, but we can also do this by FACS analysis, which now allows us to genetic screening. And this is for something I got a grant from the Alzheimer Association very recently to do genetic screens to see which proteins and which pathways are actually involved in the transport of lipids from the neurons to the astrocytes. All right, to summarize the talk, so we performed a drug screen in iPSC-derived human AD neurons, and we identified all the 42 compounds that reduce tau phosphorylation. By biochemical mapping, we show that cholesterol esters drive neuronal p-tau and A-beta accumulation to correlate with independent pathways. We are trying to clinically repurpose efavirenz as a brain-specific cholesterol-targeting drugs for the treatment of Alzheimer's disease. And we are also currently investigating whether sporadic AD risk variants, for example, in ApoE4, can drive neuronal lipid accumulation, but in a neural glial interaction-dependent manner. So if you want to read more about cholesterol metabolism as a dual driver of tau phosphorylation and A-beta pathology, I would like to refer you to our recent review that we published in Nature Reviews Neuroscience. It does not only talk about cholesterol metabolism, but also about ApoE and endosomal system as an amyloid-independent regulator of tau pathology. So lastly, my acknowledgements. There's a lot of people that I actually have to acknowledge. Cheryl, who did most of the work for the screening, Larry Goldstein for allowing me to do this work in his lab, Martin Guerra, the person that did all the lipidomics for me and with me, and the people currently in my lab, Femke Koen and Lil Eva, Matthijs Verhagen, and my clinical collaborators, Philip Scheltes, Jort Vijverberg, and Pieter van Bokhoven. So these are my funders. I hope that I get to see most of you in Amsterdam in July when there's the Alzheimer Association International Conference. And this is a picture showing that you're never too young or too old to actually start studying cholesterol metabolism. This is my daughter as 3 years already looking into that a bit more. And I'm sure she'll have a lot more to study in the future as well.
Great job, Rick. Thank you.
Thank you.
Fantastic talk. That was really nice. You've got several questions here. Some you actually answered along the way, so I went ahead and dismissed those. But if you want to see if you can pop those up and take a look. Those of you who want to take off, please feel free. Rick will run the marathon of questions here until we run out. I'll say very quick that next week we're going to pick right back up where we left off. So Dr. Heather Ferris from the University of Virginia will talk to us about astrocyte cholesterol metabolism. So I encourage you to join us again on Tuesday. All right, Rick, I'll let you take over here.
Okay, awesome. So the first question is from Jinte Middeldorp, well known to me. So did you show that other inhibitors on finalization and geranyl phospholipid indeed inhibited these processes, and whether the statins inhibit them too? Could this have any additional benefits or disadvantages for the cells patients? So we actually did not test that, so we didn't have a measure of seeing whether we affected finalization of granulation, so that would have been a good control that we didn't do. And she's also asking whether there's additional benefits or disadvantages. So I think what we showed with using the statins is that the astrocytes die, and we think that this effect actually might be dependent on the finalization or the granulation steps. But that's a very good question, and we could definitely look into that a bit more. Then the next question is which type of neurons were these iPSC differentiated to? So the way we've been doing this was a very early protocol, and one of the earliest protocols that was established in the Goldstein lab, and we don't really know what they differentiate to. So we think they're mostly cortical, but it's not a very directed differentiation. So what we're doing more recently is actually using this NGN2 system, which is a much cleaner way of differentiating neurons. And then we get a pure population glutamatergic cortical neurons. But for these experiments that we've done with the early stages, we don't really know. We think they're cortical, but we don't have the answer to that. So the next question is from Chloe. When the APP phenotype is corrected with CRISPR, is there no change in the PTAR levels? And yes, there's a change in PTAR levels. So if we reduce the copies of APP to 0, we also see that the phosphorylation TAR levels go down in these cells. And I think this is also other work from Larry Goldstein and other people that really show that this APP somehow is directly regulating tau phosphorylation as well. But the regulation by cholesterol is independent of that APP levels. Then the next question is, how did you differentiate your cells into astrocytes, and how mature are they? And again, I have to give you the same answer. So these are the early protocols that we used. How mature are they? They're not very old. We use them at 4 to 5 weeks. So they're mature in the sense that they have some astrocyte markers. For example, SOX9 we can see in there. We can see some gels with GFAP as well. But we think that these are not very mature, or the early stages of maturity of these astrocytes. So the next question is from Jenny Boucher-Prévost. Isn't it efavirenz an antiviral, pretty selective against reverse transcriptase? Is the effect on CYP64A1 an off-target effect? And which concentrations did you use in the cells? And that's actually a very good question. I had it in the slides here, but I skipped over that. Let me go back to that. Here we go. And this is, I think, a very interesting point for discussion. So As he's tried to point out, is that efavirenz was originally developed as a HIV drug, and it inhibits reverse transcriptase. And the effect of this drug on CYP64A1 seems to be an off-target event. So it's not related to its effect on this reverse transcriptase. So yes, it's an off-target effect, and which concentrations that you use in these cells. And what concentration we use is 25 micromolar. So that seems quite high in these cells. But this drug is actually very nice because it's very brain permeable and it actually accumulates in brain. So even though we have to use a very high concentration in cell culture, the concentrations that we actually use in the mice are very low. And that's also shown over here. So a normal HIV patient gets 600 milligrams per day of this drug. If we calculate what our dosing would be in a human patient based on the mouse data, we actually have to go to around 1 milligram per day for a human. So that's about 600 times lower than these patients with HIV actually would get. I think that's important because what's also shown for this efavirenz effect on CYP64A1, low concentrations actually activate the enzyme, but higher concentrations actually reduce the activity of the enzyme. So we really want to be in this lower dose range. So we calculated that about 1 milligram should do the trick. But what we really want to do in a clinical trial is do a wide dose range, going from 0.1 milligram to a 10 milligram per day range, to really see like at which dose is this compound now hitting CYP64A1 in vivo. The next question is from Kristen Onos. Do the iPSC come from lots of different patients, a small group of patients, or one patient? The genetic background of the patient also directly influences this as well. So there's another slide that I actually skipped over here. So all the work that I showed you in the presentation was actually done in this APP duplication patient. But later we went back to a much broader panel of iPSC-derived neurons, either from non-dependent controls or sporadic AD patients. And we saw that efavirenz reduced this tau hyperphosphorylation in all of these different iPSC-derived neuronal lines. So there seems to be an effect on all different genotypes. And one of the things that we're also now testing towards a more personalized approaches, using this isogenic sets, for example, for ApoE4, 4, and 3 lines that we have now, to see whether efavirenz might be more potent than, for example, an ApoE4 genotype than an ApoE3 genotype. So I think that's something that we're looking into in the future as well. Next question by Amy Chen. Is cholesterol ester independent? Is cholesterol ester dependent inhibition of the proteasome only observed in iPSC-derived neurons with hypophosphorylated tau? So maybe this also answers the question. So we don't think so. So in this case, we also see an effect of efavirenz in the non-dependent control. So we think this is a physiological regulation of phospho-tau by these cholesterol esters. But somehow this might actually be hijacked in a AD brain, for example, by ApoE4 driving the accumulation of these cholesterol esters. But lowering cholesterol esters always induces proteasomal activation, is what we observed. Constanza Cortez says, beautiful talk. Well, thank you very much. You mentioned that intercellular localization of cholesterol might be important in neurons. In addition to your lipidomic studies on iPSC-derived culture platforms, will you also be looking at lipid staining or localization? Oh, sorry, I didn't hit the answer live question. I think you can see the question now. So yeah, so the localization of the lipids is very important, but unfortunately, there's not a very good way to visualize that, at least not as I know. So if you have suggestions for that, I'd be very happy to take them on. We would be actually very interested in looking at cholesterol levels at the ER, because we think that might actually be the place where they're most bioactive in this regard. So we hope to look at that, but we don't have anything now that's ready to look at that at the moment. Question by Jill Di Paolo. He says, great talk, Rick. Thank you very much, Jill. Is there any evidence that efavirenz decreases neuroinflammation in the tau model in vivo? So we did not look at neuroinflammation in this model. That would be very interesting to look at. And I'm thinking about the experiments that Pikoleva did in the amyloid model, and I think she had some evidence that neuroinflammation went down in the amyloid model, but we haven't looked carefully in the tau model, and also these samples are still in the freezer, so we really have to look at these in much more detail still. Question by Miguel Moutinho. Great talk. 2 questions. Have you incubated these neurons with cholesterol esters or induced ACAT activity and then assessed p-tau and A-beta? So that's a very good question, and we tinkered with that for a very long time. These cholesterol esters are very poorly water soluble, so you cannot just add them to the cells to have them taken up by these cells. So that's not really an experiment that you can do. And also, I don't know a way to activate ACAT activity. We only know how to inhibit it. So we haven't done an experiment. What we try to do is feed these cells with cholesterol. And the idea was if we give these cells cholesterol, they will make these cholesterol esters, and that will induce an increase in p-tau and A-beta. However, what we observed was that if you feed these cells cholesterol, they immediately pump it out as 24-hydroxycholesterol. So particularly these neurons seem to be very well equipped to pump out any excess cholesterol very quickly. So it's been very hard for us to actually induce accumulation of cholesterol esters in these cells. So I think we have to address this with some genetic tools that we're also building now in the lab. ACAT1 ablation has been shown to increase in 24-hydroxycholesterol, which is beneficial in 80 mouse models. Could the beneficial effect of these compounds, such as statins and efavirenz, resolve around increasing 24-hydroxycholesterol? And did you measure 24-hydroxycholesterol in your treatments? So the question is, yes, we did measure that. And if we give these cells statins, we actually see that they reduce these 24-hydroxycholesterol levels. So that doesn't seem to be correlating with the effect we see on p-tau. So with the ACAT inhibition, so we also see a very strong increase in 24-hydroxycholesterol in that case. And we think it is because, again, these neurons, if they cannot make cholesterol esters, they will somehow export this cholesterol in a different way by converting it to 24-hydroxycholesterol. So I think this again shows that these neurons are just really good in exporting cholesterol. But the fact that we see that statins lower 24-hydroxycholesterol level, but also lower p-tau, would indicate it's not mediated by 24-hydroxycholesterol, but by another metabolite, which we think are the cholesterol esters.
Hey, Rick, I want to jump in here real quick and just remind everyone that we'll be back on Tuesday with Heather Ferris for a talk on astrocyte cholesterol metabolism. I strongly encourage postdocs and students to sign up for the new trainee talks. And Rick has quite a few more questions left, so I think he's probably game to continue on. So if you'd like to stay with us and listen in, please do. Thanks.
Yeah, I'm game. I'll be here all night if.
You might be. There's a lot of questions.
My wife went to bed already, so. So this is a very good question, I have to say, from Virginie. She says, "Great talk." That's an excellent remark. Thank you very much for that. So the next question from Brandon Farmer. I think I recognize that name. Lance, is that your PhD student?
Yes, it is.
Well, let's see. So great talk. Thank you very much. From the Israel et al. Paper in 2012, it looks like your iPSC neurons were from an ApoE33 individual. So that I think he's referring to the ApoP duplication patient that has a ApoE33 background, which could be true. I'm not completely sure, but I will believe you on that word. Could you speculate how your drug treatments would alter cholesterol metabolism in E4 neurons? And would you see less p-tau after statin or more? So I think that's a very interesting question, which comes back to a sort of a personalized treatment. So I personally hypothesize that these ApoE4 neurons would accumulate more cholesterol esters, and that these treatments would be more effective in these neurons. But again, that's something that we're testing at the moment, so we'll know soon enough. Question, apologize if I missed it, but was the neuron astrocyte co-culture in trans-well format, or was it direct cell contact needed? So the answer to that, it's both, I think. So I need to think, because the Femke did those experiments. I think the data that we showed was where we have a plate of neurons, and we put a couple slips of the astrocytes on top, but they're not touching. So we make we make a barrier through plastic tips where they're separated from each other. So they, I think we separated them. They were never in direct contact. I see you pulled up Femke, so she might actually have a better answer to that.
Yeah, in this case, they were indeed not touching each other. So it was a co-culture with two separate cover slips, which were separated from each other, but in the same medium.
Good. So that means that I'm not such an advanced PI as I would like to be, because I still know the experiments being done. So that's something I have to work on. But thanks, Femke. So someone thanking me for the lecture. Thank you for the comment. Next question is, when you looked at total cellular cholesterol levels in the statin experiment and observed no changes, did you look at subcellular distribution of cholesterol to see that that perhaps changed rather than total cellular levels? And then that's a great question. So we didn't look at that, and we would be really interested to study that a bit better to see if there's maybe a localized pool of cholesterol that's actually regulating the proteasome in this system, and that would be a very good starting point to see how this is regulated. So something we haven't done, something we would like to do. I just don't think that the tools are there yet to really look at that. For example, Philippine staining is not, I don't think it will be sensitive enough to detect local changes in cholesterol levels, but this is definitely something that we're interested in looking in the future. And next question, do you know if efavirenz treatment affects oligodendrocyte production and myelin levels or ultrastructure? No, we don't know that. We haven't tried that. If that's something of interest to you, I think that's something that we could definitely test as well. We don't have the protocols to make the oligodendrocytes in our lab, but you can also make them from the iPSCs, of course. The next question is, I'm not aware of it in detail, but do iPSCs not lose the aging phenotype when we program typically? And do you control for this in any way? So I think the answer to this is generally yes. So they don't have an aging phenotype. They are young neurons. So we really use them to look at cell biological processes downstream of sporadic AD risk variants. And I think aging is always something difficult to define. And one way you could define aging is by increased exposure to reactive oxygen species. So one way maybe introducing aging in this system is actually by giving them reactive oxygen species or impairing mitochondrial respiration. So those kind of treatments I think might actually be relevant to the system, and they will also induce these neurons to produce lipids a bit more. So we don't control this in any way, but we think that certain aspects of aging could be involved in the mechanism as well. The next question is from Nadia Morshed. Did you explore any post-translational regulation mechanisms on cholesterol ester producing enzymes? So if I understand the question correctly, whether I looked at post-transcriptional regulation of genes that produce cholesterol esters, which would be the ACAT enzymes, and we haven't looked at that at this point, but that would be interesting to do for sure. And the next question is from Marcus Lee. iPSC-induced astrocytes ApoE22, 33, or 44 have different levels of lipid droplets. Question. So again, we had the astrocytes ready. We had the whole series fixed. Femke was ready to go in and do the stainings, but we got shut down by the COVID situation. So we haven't looked at that in our human iPSC systems. But Lens, I think you have looked at that in the mouse system, and I think you showed in the last talk that actually the lipid droplets, amount of lipid droplets, are different in astrocytes from ApoE22, 22, 33, and 44 carriers, if I'm correct.
Yes, at least in the mouse model, we think so based on the preliminary data. The E2 are a tricky situation though. That mouse model is hyperlipidemic. And so if anything, they appear to have more droplets as well. So I'm not sure the translational value there with the 2 2s, but the E4 mice at least look like they have more.
Yeah, so this is something that we'll definitely test in our astrocytes as well, and we'll also do the lipidomics to see really which lipids then are accumulating in these astrocytes. Next question, efavirenz showed no toxic effects compared with other drugs. If you could go to that slide, I would appreciate it. Yes, I'm very close to that slide. However, the number of initial astrocytes was less than the numbers of neurons compared with other drugs. Why did this happen? So I'm not sure if I understand the correct question completely. I'm not sure if I understand this question. So I don't think there was a difference in the amount of astrocytes when we started this experiment. So maybe if you can rephrase the question, I can come back to that later on. So maybe Lance, can you help me? Do you understand the question better?
Yeah, I don't. I thought maybe it was referring to the viability, but that no, I'm not sure. Sorry.
Oh, can you find Johnny and unmute him? Maybe you can ask his question if you want. So. I'll go to the next question. Okay, there he goes. Yeah.
Hi, can you hear me?
Yep.
Yes, we can. Yeah.
Well, thank you very much for the talk. It was really good. And yeah, I was referring about viability, because in simvastatin, the viability of astrocytes seems to be higher than the ones of neurons compared with efavirenz. I'm not an expert in that, so I just wanted to know why did it happen and which potential effects could it have.
Okay, I see. So you're saying that this red line over here starts at higher than 100%. That's true. So that actually would mean that low doses of efavirenz would enhance viability of these neurons, and that's the neurons, yes, but not astrocytes. Yeah, so that's actually an interesting observation. We hadn't seen that before. So thanks for pointing that out. And I think that's something that would be interesting, because that would indicate that low doses of efavirenz also enhance even enhance neuronal viability in this system. So yeah, very good point. Thanks for clarifying. A question from Irene. Do statins cross the blood-brain barrier? You said that there were very reduced levels in the brain. So some statins do cross the blood-brain barriers, for example, simvastatin, but they don't get into the brain at very high levels. So they do reach there, but not very efficiently. Next question about the transfer of lipids between astrocytes and neurons. You show accumulation of lipids in astrocytes when neurons are coated in the same well. How specific do you think this process is? Could it be that astrocytes phagocytose neuronal debris that contains the lipids? Yeah, so that's a very good question. So that's we don't know at this moment. So the work from the Bellon lab actually showed that they're specific and ApoE dependent. And we're trying to see now if we do the same experiment, for example, an ApoE knockout background, if we don't see this transport happening anymore. So that's one control we want to do. And also there's a way to block this export of these lipids, lipoproteins, by inhibiting the export from the Golgi, but by refeldin A. And that's also a way we want to see if this is really specific to like lipoprotein export from the Golgi to the neurons, like from the Golgi in the neurons to the astrocytes, or whether it's just debris. So we haven't done a proper experiment yet. We're doing it at the moment to control for that. Then we have a question. So what's the chain length saturation of your cholesterol esters? Yeah, so I'm not as familiar with the fatty acid chain lengths in these cholesterol esters as I would like to be. Maybe I can look back at that data. I don't think we saw a very clear difference between the length. So I think it's both the. That's true. I have to look back at the data. I can't answer that now. If you send me an email, if that's something that you're interested in, I can definitely share that data with you as well. The next question: given the high variability in iPSC cultures, do you expect to see similar drug screens result in results if performed in more patients. So late onset patients are, as opposed to the ones carrying highly penetrant APP mutations. I think that's a relevant question, and so what part of the answer is that for the statins and efavirenz, we have been able to repeat that in any cell that we've been looking at today. So I think that result is very replicable. It's also something that's targeting lipid metabolism, which I think is both changed in both the sporadic AD patients, but also in the familial AD patients. If you look at the mouse data, if you would do the entire drug screen again in a sporadic AD patient, I think you might get different hits, of course, depending on the mechanism that's upstream of that. So I think it would be very, very interesting to compare such screens also to see if you can also get some hints about the mechanism. A semi-related question: given your background in endosomal recycling system, what do you think about papers indicating that ApoE4 cells have malfunctioning endosomal cycling because of their pH differences? There's some evidence that histone deacetylase inhibitors can correct this. Any evidence that these inhibitors are useful in your current models? Thank you. Yeah, obviously I've seen that data. I think it's from a very good lab, and it looks like good data as well. So on face value, I would say that's it's true. We haven't done any. I don't think we had any hits that were histone deacetylase inhibitors in our screen. But they can also look at the specific compounds that no, I think SAHA actually came up as one of our hits. So yes, SAHA came up as one of our hits. So it could be an indication that this system is also involved in our system. Again, a question from Angels, my old colleague in the Goldstein lab. Besides changes in phospho-tau proteasome-dependent degradation, have you looked at the effect and degradation of other relevant proteins, or is it tau specific? That's a very good question that we're very interested in also. So there's been a couple of papers that recently showed that ApoE4 also, for example, drives the accumulation of alpha-synuclein. TDP43 is also something that's the accumulation is driven by ApoE4. So we hypothesize that this is not specific to tau only. This is a broader regulation of the proteasome, but that certain proteins are just more prone to accumulation than other proteins. So we don't know at this point whether it's tau specific, but certainly something that we're interested in and looking at. I have to say, when we did the proteomics, it's not that there's like 200 different proteins that are differentially regulated. So there seems to be some kind of specificity. We just don't really understand where that comes from at this point. Next question is, are you planning to perform lipidomics experiments in neurons and astrocytes only, or also in co-cultures? And are you planning to target non-coding variants as well? So yes, the first part is yes. So we're going to do microglia co-cultures with neurons as well. Femke is also working on those systems. We want to make all the different combinations. And are you planning to target non-coding variants as well? At this point, we don't have plans to do that, mostly because we like the coding variants because we think they have the strongest effects. We also know how to make them by CRISPR, but if there's any certain non-coding variant that you're interested in, I would be very interested in looking at that as well, of course. The next question, I was wondering which patients you were selecting for your clinical trial with efavirenz, and if you would select on certain CSF levels of p-tau-tau. So we're going to do it in MCI patients, so early Alzheimer, no, MCI and early AD. I think that's what we decided on to do. We're not going to stratify them by ApoE4 status, so we're just going to take all the same patients. And I think they get the diagnosis based on A-beta levels. So definitely they have the criteria of certain A-beta levels to be included in the clinical trial. We're not going to be looking at phospho-tau levels at this point. And I think it's important to mention, so at this point, we only want to do a very short clinical trial see which dose is most effective in activating the enzyme. And we only want to treat these subjects for maybe 4 weeks. If we find a dose that's effective, we're going to move to the next phase, which might be a phase 2 clinical trial. And I think then these questions will become more relevant. And you would like to, I think, have patients that have high phospho-tau levels in CSF or even high tau PET by MRI, by PET scanning. And the next question is just moving a little bit from cholesterol metabolism. You briefly mentioned SORLA for the SAD risk analysis. Do you have any preliminary data regarding this? Because it's known that SORLA increases lipid accumulation in adipocytes through increased insulin signaling, maybe neurons, glia as well. On the other hand, if it decreases amyloid load in the AD brains, which aspects of it are you guys interested in? So that's a collaboration I'm doing with Jessica Young in Seattle, and I can say she's very much interested in the effect on amyloid load and amyloid production, and together we're also looking at lipid levels as well. So we'll look at both of these the questions that you asked, but the data that I have is really too preliminary. It's from one experiment, so I hope I can repeat these measurements soon, and then we have some more idea about these effects. Next question is by Garrett Howell. Excellent talk. A more general question: does lipid droplet accumulation spread in the brain the same way as tau pathology is now thought to spread? Yes. I don't know about spreading. I mean, the concept of spreading is like prion-like spreading, maybe. I don't think it will work like that, where you have like a seeding of one lipid droplet somewhere else. But obviously, if there's lipids that are being secreted by neurons, they can travel to different brain areas. So there must be, I think it would be more like a gradient than really like a spreading, like a prion-like spreading. I think what's really interesting is that, for example, proteins like ApoE and TREM2 are really highly expressed in the EC and parts of the hippocampus. So I think it might be more depending on the expression of the proteins involved in this process than like a prion-like seeding form. In your initial drug screen, I noticed that berberine seemed to have a strong response in terms of cell viability and p-tau inhibition. Did you ever spend any time on that? Or do you have any thoughts on berberine and its related pharmaceutical compound metformin? Yeah, that's an excellent question, because I actually have quite a story also on berberine, and I was doubting whether I should show it here or not. So as you notice, we see that berberine also reduces tau hyperphosphorylation in these cells, and the cells are still alive and happy as well. So we're looking into that, how that works, and we think that might be a process that depends on AMP kinase and the mTOR pathway. But it's something definitely we're trying to wrap up a paper on that, and so I hope that will be out hopefully within the next year. And so your metformin comment, so this berberine is an inhibitor of complex 1, and that's also something that metformin does. And metformin, of course, is now in the clinical trials for Alzheimer's disease, phase 3 clinical trials, I think. So indeed, we think it's a very similar mechanism for both of those drugs. The next question: have you looked at other familiar AD mutations such as presenilin? We haven't. So I think the question is, have you looked at lipid levels in the presenilin carrier? So we haven't looked at that. I think that would be something something interesting to look at. I'm currently not working on a presenilin model in my lab, but Larry has a couple of these mutations in his lab, and at some point it might be interesting to look at those as well. And then I think this is the last question from the series. From Alicia Nugent, so I'm assuming that's the first author from the cholesterol ester paper in the microglia. Since you see p-tau reduction with efavirenz in the non-demented controlled genotypes, is there any concern that changing physiological p-tau levels could be detrimental for normal tau function? So I guess the question is, would you treat non-demented controls with efavirenz? And I think you would only go into patients with FF variants when, and in that case, you would have already increased p-tau levels. I don't think there will be a negative of FF variants, a negative effect of FF variants in these non-dependent controls, but I don't see why we should treat these patients with FF variants. And the second part of the question, does FF variants also lower total tau levels in NDC neurons, as I believe you showed in the APP dupe iPSC lines? So we haven't looked at that in the non-augmented controls, but I would guess it would also reduce total tau levels there, because I think the mechanism that we see in general is very similar, whether we do this in non-augmented controls or in APP duplication or in sporadic AD patients. So we think the mechanism is all converging on the degradation of tau by the proteasome. So I would expect a similar effect over there.
Hey, Rick, I have one more question for you.
Yeah.
So did you see any effects on other post-translational modifications of tau from the statins or effervescent, such as acetylation of tau or ubiquitination of tau?
Yeah, that's a very good question. Also, a question that we haven't looked at that, obviously. So I think the way you would have to do that is by doing an IP on tau and doing a mass spec, and I don't think I have the facilities to do that at this moment, but I think that's a very interesting way to look at it. And I think, especially in the early time points, maybe that would be a first change in tau that then induces their degradation. So that's something that would be very interesting to look at for sure. Yeah.
All right, Rick, great job, man. Thank you so much. We had over 270 participants, almost 70 of which stayed through your marathon of questions. So great talk. Thank you. Thank you very much.
All right. So we'll be back here on Tuesday.
That's right. All right. See you guys then.
Series, I think.
Great. All right. Take care, everybody.